Okay, so thank you very much, Mr. Chairman, for this kind and exhaustive presentation, introduction. I would also like to thank the organizing committee to give me the opportunity for this presentation, and Ronaldo for organizing all this. So I'm working with Ipsen. It's a Paris-based organization, company working all over the world, let's say 1.5 billion turnover, focused on peptides and toxins, and I would say working on delivering treatments for patients in many different diseases, oncology, cancer treatment, but also endocrinology and neuromuscular disorders. So there's a strong focus in Ipsen on peptides, and as you know, peptides are amazing drugs. I would say the missing link between the monoclonal antibodies, quite complex to develop, and the small molecules which are more toxic. So a lot of advantages, but also I would say they need to be administered chronically for long periods of time. Most of the time by injection, and so there's I would say, strong interest in developing alternative routes of administration, and I'm going to focus on the oral route after describing the landscape of peptide delivery at the present time, and I'm going to focus on technologies that are already progressed in the clinic, and to also talk about the future challenges. So let's talk about peptide delivery landscape. So as I already mentioned, these are drugs that are mainly administered through injection, but also, as you can see here, these data clearly show that there are also some peptides that are administered through topical, nasal, or oral route. And well, if we have a look here, I don't mean to be exhaustive, but this is a table where you have, I would say, most of the peptide forms that are on the market today. And as you can see, most of them are sustained-release formulation, injectable sustained-release formulation from 1 to 6 months using various types of technologies, microspheres, implants, and so on. But you can also see that you have some nasal spray formulations that are on the market at the present time. So here is a, I would say, technology trend presentation. So as I mentioned, the most common form is the injectable sustained-release formulation using many different technologies, microspheres, implants, self-assembly technologies, which are the most recent ones, and people are focusing now on developing ready-to-use formulations in prefilled syringes, and also using narrow gauge needles, focusing on, I would say, the patients for compliance and convenience. The other route that is also quite investigated at the present time is the transdermal peptide delivery, where you have products in phase 2, for instance, PTH analogs, by, for instance, 3M working with a radius in this area, but you have also other companies working in this field. Then there's the oral peptide delivery with a broad range of technologies, and this is actually where we are going to focus now. It's clear, and as you have maybe heard, there's a octreotide form oral that has been filed in the US very recently by Chiasma and should be approved this year. And there's a strong competitive pressure, as we will see. And the last one I would like to mention, just for the interest, is the intracellular peptide delivery, which is quite challenging, addressing, I would say, targets for cancer treatment or brain disease treatment. So we are now going to focus on oral peptide delivery. And first thing I would like to mention is that there's a strong expectation from the market. There's a strong unmet medical need, and if you, I don't know whether there's a pointer here somewhere, no. But if you look here at, I would say, patients that are getting the injectable forms, most of them say that they would prefer the oral form, even if it would be BID. So I think it's a quite interesting statement. The other aspect is that, and this is something that has been already investigated and even developed commercially, it's the possibility to deliver to a site of action in the GI tract. And this is, for instance, what was achieved with oral linaclotide, Lince's product, targeting receptors in the GI tract with actually two mechanisms of action, one local and one systemic, acting on nociceptors. And, well, I would say there's a strong competitive environment, a lot of money put in clinical trials, and also statements about, well, if the octreotide capsules from Casma are approved, this will be the first oral somatostatin analog, and this will be quite an achievement. But there are also other products that are, I would say, quite developed at the present time. These are GLP-1 analogs, and Novo Nordisk is very active in this field, as you may imagine, spending a lot of money in this area. So first, I would like to mention that we already have marketed oral peptides. Here you have a list. I do not pretend to be exhaustive. These are eight of them. Most of them are for local action in the gastrointestinal tract. And most of them also are cyclic peptide, as you can see. Thank you very much. Okay. So most of them are cyclic peptides, and you can see that, well, there are just a few of them, like for instance Ceredis, which actually is acting centrally, and I think it's very important because it demonstrates that you can get an active, efficient, efficacious product, and also from an industrial point of view, financial viability of a product, which means that you are able to cope with the low and variable bioavailability of a drug. So if we look at the challenges, well, as you know, peptides are poorly absorbable through the GI tract. Whatever the transport mechanism, it can be a paracellular mechanism or transcellular mechanism, and if you look at the ratio of the surface of a tight junction versus the cells, of course, the cell seems to be much more favorable, but going through the cell might be a little bit more difficult. So actually you have to find the right balance. And so there's an opportunity to target receptors in the GI tract. We have already mentioned this, for instance, for IBS, and this is what Lince is doing, by the way. But actually what we would like now is to increase the absorption for systemic effect, and there's then a need for permeation enhancers, which are novel excipients or nanoparticulate approaches. And if you look at the literature, you can find a wide set of technologies investigated in clinical development. Here again, I do not pretend to be exhaustive, but you have here quite a interesting list of different technologies, going from, I would say, kind of physicochemical interactions with lipophilic transport complexes, but you can also have nanoparticles or micellar solutions, or also having some covalent complexes with amphiphilic oligomers. So it's well, I would say, a very active field of research at the present time. And it's just a summary. And I would like just to mention here a few classes of, I would say, technological approaches. And the first one is the absorption enhancers, because actually going through the membrane is the main challenge today. And here you have a list of various absorption enhancers that are used in these formulations that are now in the clinics, which means also that they have gone through all the toxicological studies. You have also enzyme inhibitors, because the other key challenge is the stabilization of the peptide in the GI tract, and you have to avoid enzymatic degradation. There are other ways of doing it, like using, for instance, enteric-coated dosage forms, capsules or tablets. The other aspects are related to mucoadhesive systems, because actually you want to keep the drug close to the absorption locus as much as possible to increase the time of residence in the GI tract. Other aspects are related, for instance, to increasing also going through the membrane, passage of the membrane through conjugates, for instance, using cell-penetrating peptides or bacterial toxin conjugates, Pseudomonas exotoxin A, for instance, developed by Trinity Biosciences, or other conjugates. And also there are approaches developed using functionalized nanoparticles, but they are not in the clinics as much as I know. So the most commonly investigated and successful approach is the one based on absorption enhancers. And here, for instance, is a well-known technology, peptiligence, which is contraction of peptide intelligence, I would assume. So going in phase 3 with other products in phase 1 to phase 2, calcitonin, PTH, and so on. Many partners, full development and clinical manufacturing capabilities. Another one is developed by Marion, and here, as you can see, there are various stages of development of the technology with various, I would say, evolutions of the technology, going from a first generation technology of powder of API in enteric-coated tablets, then going to microemulsions, and then to mixture of fatty acids in enteric-coated gel capsules, so quite developed formulation. And, well, I would say that, well, it has been shown to have a strong interest for oral insulin, and actually Marion they have sold their asset to Novo Nordisk end of 2015, which I think is also quite interesting news in terms of the, I would say, potential success that Novo Nordisk is expecting from the technology. So let's just have a look at the nanoparticles, and I would like more particularly to focus on Fc-targeted nanoparticles, because I think this is a quite interesting technology that was published a few years ago. So these are nanoparticles obtained by nanoprecipitation from D-block copolymers, PLA, PEG with a maleimide functionalized group. They can be coupled to Fc fragment of an antibody, which is quite interesting, using the reaction with thiols. And as you can see, the diameter of a nanoparticle is not, or almost not, modified by the functionalization. The density of Fc fragment is quite high. And there's in vitro, there's a very interesting effect of going through membranes. So it's quite interesting. And then if you go in vivo, you can see here the proposed mechanism, which is about well, a transcytosis pathway, and then exocytosis with a pH-mediated dissociation. At the surface, initially, you have interaction of the Fc fragment with a receptor, neonatal Fc receptor, which makes it possible to recognize the cell, and then to have a kind of active transportation mechanism through the intestinal epithelium. And as you can see here, there's a strong enhancement of absorption that is related to the functionalization. And of course, you can see here that versus the non-functionalized nanoparticles, you have a strong presence of the nanoparticles. So I think it's a very promising technology. So now let's focus on the technology's progress in the clinic. So here, once again, this is not exhaustive, but you can see that you have products that are close to filing. Other ones are in phase 3, octreotide, calcitonin, insulin, or GLP analogs, or the products that are the most developed at the present time from phase 1 to filing. And, well, as you can see, most of them use today absorption enhancers. They are based on various dosage forms, either enteric-coated capsules or tablets, but you can see that most of the time you have absorption enhancers. Some of them are based on nanoparticles, gold nanoparticles in phase 1, for instance, which is quite interesting also, as well as calcium nanoparticles. Yeah, I would like to have a focus on this one, which is a little bit a new one in the field and a paradigm shift. Actually, this is an intra-enteral injection. This is a combination of oral absorption, but then microneedles that inject in the wall of the intestine, as you can see here. So there's a quite smart system with a inflatable balloon that makes it possible to press the microneedles against the wall of the intestine and then to deliver the drug in the wall. So it's enteric wall delivery of a system. And actually you have two teams working in this field with a strong interest also from industrial companies, like for instance Novo Nordisk, Novartis, but also AstraZeneca. It was published last week. So I have a strong interest in using this type of system to deliver drugs, high molecular weight drugs, peptides, and proteins by the oral route. So now, a few words about what do we learn from the clinics and how the companies have addressed the challenges of peptide stability and oral bioavailability. And the first point is, I think it's quite interesting, is that you can achieve similar pharmacokinetic by the oral route compared to subcutaneous injection. And this was shown, for instance, by Chiasma with the Octrolin product in phase 1 studies. They also showed food effects and also interactions with proton pump inhibitors. So these are maybe aspects that have to be taken into account. But you can see here that a 20 milligram oral octreotide achieves similar PK compared to 0.1 milligram subcutaneous injection. You may be amazed by the ratio. It's a 200 ratio, which means bioavailability is still very low, but it's still considered to be workable and susceptible to go to the market. There are food and proton pump inhibitor interactions that decrease the bioavailability. The other thing is that orally delivered peptide remains bioactive. And this is also shown here in this very nice study on octreotide, where the orally administered octreotide has a very good control when the challenge is applied. So there's a GHRH arginine challenge, which is applied after 2 hours, and the blue curve shows the control with the oral octreotide versus no octreotide. So you can see that there's a very good control. So octreotide is used to decrease the GH secretion. It's used in acromegalic. So these are people that secrete too much growth hormone, and so you have to control the secretion of growth hormone, and this is exactly what is done here through the oral route. The other aspect that was also learned from the clinics is that oral delivery may also lead to increased efficacy. And for instance, this is here a phase 3 study carried out for oral calcitonin versus placebo and marketed nasal spray. And as you can see here, the oral calcitonin provided a greater increase in the lumbar spine bone mineral density, so which is I would say the marker for this type of drug and disease. And it's clearly shown here versus the nasal spray and the placebo. It was also shown to be very safe and well tolerated. Unfortunately, at this time, the agencies did not recommend to pursue this type of drug in osteoporosis. It has changes recently, but this is why this study was stopped. And Another important thing also I have already mentioned is that there are potential unexpected food effects. And here, for instance, this is another oral calcitonin with hemisphere technology, and they showed that the absorption depends on the volume of water which is used to administer the drug, which is quite amazing, and also on the time before the meal. And so, well, I mean, you have to deal with this, and you may have some specific indications in your instruction for use, but this is to be taken into account. So, before the conclusion, I would like to have some thoughts about how can we anticipate the switch from injection to the oral route. And the first statement is that relative bioavailabilities for oral absorption will always remain quite low, except maybe with a new enteric injection that has been developed, for instance, by Rani. But, with the state-of-the-art technologies in the clinic today, it's quite low. Dosing frequency may need to increase, 2 times per day, 3 times per day, why not? And then you have to balance, are the patients ready to take pill 2 times or 3 times per day versus an injection every 3 months or 6 months? So, well, it is something to be put in the perspective. Cost of goods will likely increase a lot because actually you will need a lot of API. But to counterbalance this, there's no need for aseptic manufacturing of a final dosage form because actually this is oral and no longer injectable. The cost of API may decrease as production scale will increase, and maybe you can get an upside in sales. But, well, this is a maybe. You will need to invest a lot to increase your capacity for API manufacturing because otherwise you will not be able to produce the, 100 times more API that you will need. And in addition, last question, but not least, will the oral treatment be reimbursed? It will need to demonstrate superiority versus the reference treatment with potential increased safety and efficacy. And, another consideration is that if you consider competing technologies for the oral, like transdermal, nasal, or inhalation, actually, the oral route offers the lowest bioavailability for peptide delivery compared to alternative routes. The other ones can reach 20%, 30%. But oral will still remain a few percent, I would assume. So before going to the conclusion, so the key changes as a summary is, first, bioavailability and variability versus the other routes, it's clear. Peptide stabilization, we have not talked a lot about it, but, it seems that various academia groups and industries have found quite interesting ways using enzyme inhibitors or over enteric coating to stabilize the peptides, but there might still be some challenges to come down the road. The other point is the absorption and enhancer safety. You have to carry out all the toxicological package for this absorption enhancer.